Well, in any case, I'm very pleased to be able to say a few words about Akira, although Madison only asked me yesterday to do it, so I didn't have a heck of a lot of time to prepare something. So I'm going to have to sort of work off my general memories, because when I came here, Akira was already a treasure to the then Marine Sciences Research Center. He was a very humble person, a very quiet person, and yet an extremely powerful and influential person, because so people had deep respect for him as an individual and also as a phenomenally talented scientist. And he never promoted himself, even though he was a tremendous leader in his area. He was there, and he was incredibly important to a whole range of different people, and in particular in the disciplines of physical oceanography and in ecological theory and the study of physical and biological interfaces in the environment. And so this is actually a drawing, I think you've already seen this, from a special issue in Oceanography, the journal Oceanography, in 1999, that was a tribute to Akira, who had died in 1996. And a number of people contributed this, eminent scientists at the time, and also many students who cared deeply about Akira and the impact that he had on their lives. And I urge those of you who have an interest, in fact, I urge you, if you don't have an interest, you should have an interest in looking at this issue of oceanography and reading about him, his biography and his impact on people. And so Malcolm Bowman normally would give this introduction. You can see he had a long history of interaction with Akira, and we used to, in Marine Sciences Research Center, when Jerry Schubel was dean, virtually everybody got a plaque or something. There was some kind of event, and Jerry would hand a plaque. And I don't know where the plaques were made, but everybody got a plaque, and Akira got a plaque in 1995 in honor of his many contributions to the Marine Sciences Research Center. And it was a great thing. Now, I learned first about Akira due to his contributions in physical oceanography. And there's a famous paper called Oceanic Diffusion Diagrams by Akira in deep sea research in 1971. And those of you who study turbulence and eddy diffusion processes in the ocean are probably quite familiar with this early contribution to try to look at some of the tests for fundamental theory of turbulence and eddy diffusion. And so this is called an Akira diagram. It plots the eddy diffusion coefficient in the horizontal plane versus the scale, the length scale at which you're observing the transport process. And there's a clear correlation here, and it has a basis in similarity theory, which those of you who know about it know what I just was talking about. Those who don't will have no idea, and I'm not going to go into it. But basically, this was a contribution, and I stumbled on this. And in my own research on looking at bioturbation in sediments, I wondered whether a similar kind of correlation could be found in terms of mixing of sediments by organisms and the length scales, the sizes of those, which I won't get into further here. But he contributed very fundamentally to these kinds of theories, and he wrote a very famous book that was published in 1980 that crossed over many boundaries, very interdisciplinary in its characteristics, looking at the physics of transport and diffusion, and both physical and biological processes that could be examined using that theory. So he was he had a very high stature as a scientist. And Simon Levine, after Akira passed away, put together a follow-up on that earlier text, which was published in the 1990s. And you can still get this on Amazon for $80 or so, I think, or you can rent it. for much less. And I think it's fun to look through this. And so many people who influence the marine sciences here at Stony Brook can be seen in this picture. There's Don Pritchard, who was the advisor for Kira when Kira got his PhD at Johns Hopkins. And in his own right, Don Pritchard was a very famous estuarine physical oceanographer, and you're probably familiar with this paper. There's an award given in his honor by SURF still. And there's Bob Wilson, now retired. He already had gray hair back in those days. So I should say a few things, because to show you his stature in the field, there's a list of people who came and spoke at the symposium. And many of you knew some of these people personally, but they were giants at that time that they came here. So he was extremely respected in science, physical oceanography, and ecology, and interdisciplinary. Now, one of my memories about Akira I should relate. So we used to go to dinner with speakers, and he would always come to dinner with speakers. And he was, of course, Japanese, and he loved things like raw fish, sashimi, and that sort of thing. And one speaker, I remember we went to the Three Village Inn. It used to serve traditional New England dinners and so on. And Kira looked for a while at the menu, and then things like clam chowder and that sort of thing. And he ordered the tuna that night, blackened tuna. And out came, as part of the meal, out came the blackened tuna. And he looked at it for a while, trying to figure out what it was. And that it was really tuna, and it was extremely well done. I can tell you that. And I don't think he'd ever had tuna that was cooked anything like that. So he looked really, and but he ate it, or he tried to, and he's a good person. And the other memory I would relate to is that both of us used to bike into the university all the time. So in those days, there were in the 1980s, there was no bike path here at Stony Brook. And so you went up the road, and we were going up the road past Engineering Drive there, and which is a hill on Circle Drive. And Akira would sort of, I can tell you, a little bit like diffusion, like the process that he studied. He would kind of weave in the roadway as he went up the hill. And I was much younger then, so I would just race by him and sort of leave him in the dust. But he was really, it was just wonderful to see him do that every day and remain active right to the end of his life physically. So I'm just gonna leave it there. Just a few words about him and the general role he played here in making this place a really intellectually inviting place to be. And so I want to pass this now to Nathan, and hopefully some of you get a sense of how important Akira was in the life of this place.
So before I introduce Dr. Brugler, I wanted to say thank you all for giving such a warm welcome to him. He's had a great time so far. I think many of you enjoyed his really phenomenal talk yesterday. And I also, on behalf of the GSC, I wanted to thank you all for making this event possible. We can't do this without all of your faces. And also I wanted to thank you for your participation in the fundraising events we have. We do a Okubo Fest and the bake sale in the fall, and all of that money goes towards bringing him here, picking him up from the airport, housing him in the inn on campus, which he says is fantastic, and giving him all a lot of great food, which I know some of you are going to enjoy tonight at the dinner at Grumpy Jack's. And so, I know many of you have already met him. I'm not going to read him his bio. You've gotten way too many emails about it already. But I'll introduce Dr. Mercer Brugler from the University of South Carolina, Beaufort. And I'll let him take it away from there.
Cool. Thank you. Good afternoon. Thank you guys for being here today, and thank you, Graduate Student Club, for the invite. Where to even begin? I used to be a deep sea biologist, but ever since moving to the Low Country in South Carolina, I'm getting a lot of pressure to study shallow water stuff. So we'll see some of that throughout today. So yeah, currently at the University of South Carolina, Beaufort, got there in 2020. Although my paycheck is being paid by a private foundation right now, still affiliated with the AMNH as well as the Smithsonian in DC. They've been great about providing funding for research projects and students, which I'm very thankful for. Thank you. Thanks for saving the day. All right, so the title I realize is a bit funny, maybe not professional. My current shiny objects, but again, my mission is to change the face of science, to specifically recruit underrepresented minorities. And in doing so, I need to retain them. I need to inspire them. And so I've really diversified my research portfolio to do just that, to inspire the students. And so I say shiny objects, really it's the students that are dictating what we're studying at any given point in time. So this is kind of where we're putting most of our efforts right now, from deep sea corals to oyster reefs, to horseshoe crabs, to leeches. It keeps everybody having fun. Oh my goodness.
I think it's just
Not full screen. All right, let's do.
it's not like just like, yeah, now just go.
All right. Sorry. I'm so sorry. I promise I've presented before. I promise. First and foremost, like yesterday, I want to thank Roy Price. Without him, I wouldn't be connected with you guys. I'm very thankful for him. Met him at a NASA exobiology review panel in California, and we hit it off right away. So very thankful for Roy. Love this guy. Also thankful for the Graduate Student Club. Thank you guys very much for this honor. It's absolutely awesome. In fact, my boss called me yesterday. He's like, "So, how's it going?" I'm like, "It's going really well." He's like, "So we should talk about full professorship." I'm like, "Why are you calling me right now about this?" Anyways, I thought that was interesting. Again, very thankful for this individual. I've learned a lot about him, not just through reading this tribute in oceanography from '99, but speaking with you guys and seeing his setup in the library, his chair, table, lamp, and books. I think that's pretty cool that you guys preserve that. So, and I ran in, I'm rather tall. I ran into one of his signs in the library, literally. I saw a tree was dedicated to him, the conference room. You guys are really honoring this guy, and it seems like he was an amazing individual. So very thankful for him and his contributions. I also want to thank my students before we get started, because everything you're about to see, and this isn't data heavy. I'm trying to inspire you like I'm trying to inspire them. And I don't want to get lost in any weeds here, but I'm very thankful for the students that are driving all these projects. Jacayla, all three of these have just left the lab, three of these individuals. Jacayla's at a PhD program at the University of Alabama Dolphin Marine Lab, but Aisha and Yessi are currently in limbo looking for a suitable PhD program. So I just want to throw that out there, but they're the brains behind everything that you're about to see. I'm not going to go through this. I'm going to start with, this is kind of my academic trajectory. I'm going to start with in 2010, I came to New York City and started a postdoc at AMNH. 2014, started at New York City College of Technology, my first tenure track position at the J Street Metro Tech stop. And along that same time, I started adjuncting or doing lectures at City College, NYU, and Columbia, while also teaching at CUNY. Absolutely loved my experience at CUNY. But nonetheless, in 2020, I decided, well, before then I started applying. The rat race in New York City, my family was growing. I was having way too many kids. And I was spending more time commuting than I was with my family. And at that point, I was like, I've got to focus on my family. So I started applying elsewhere to marine labs around the country, particularly along the coast. This one hit. I've been looking for a way back to the Low Country since I went to the College of Charleston for my master's. Just love that marsh environment. So here is just to show you. So we've got Charleston, South Carolina, Savannah, Georgia, Hilton Head here. So we've got three campuses: Beaufort, Bluffton, and Hilton Head campus. I'm based in the Beaufort campus. One of the reasons I love this particular area is because we have 8.5 feet tidal surge every 6 hours, which is crazy. And so here we are along the coast here, and we're the deepest natural harbor. And I don't know if that's why we have so much water coming in and out, but you can see the continental shelf and slope is a little funky down here in the South Atlantic area, South US. While I'm parked here, I I wanted to use this to show you the bathymetry of the area leading up to where I am, but also say I've submitted this grant to NSF 3 times and I've been rejected each time, driving me absolutely nuts. Again, speaking to the failure aspect that we were talking about yesterday. What are we trying to do? Well, Eric Cortez from Temple found an 85-mile-long Lophelia coral reef at 800 meters depth there at the Charleston Bump. And we're trying to get money to go back out and study it. We've been unsuccessful to this point. So wanted to bring up some more failure on my part, but keep going, keep persevering, keep putting in those proposals. So this is what it looks like in the Beaufort area. We're dominated by Spartina alterniflora, the smooth cordgrass, or Sporobolus alterniflorus, as it was reclassified, but very much dominated by Spartina. Again, big tidal amplitude. This is the view from my office. We're right on the water, small marine lab. We've only got 2,000 students across all 3 campuses and about 90 students in the undergraduate marine biology program. We do not have graduate students, and I love that because I can mold those undergraduates. They're still trying to figure out what they want to do. It's like the perfect time to get them interested in the marine environment. Here's my house. I realize that real estate is a lot cheaper in the South. So I've got a house right on the water, putting in lots of trees to buffer. I can't believe how close the house is to the water. So trying to buffer it with trees, but I'll have a shallow water dock. And so all of this clears out during low tides. This is all pluff mud. So I can only get the boat out during high tide. Spring and king tides bring about 10 feet of water in there. But the reason I went after a waterfront property is so I can bring all of my classes there. So there's lots of opportunities to bring students into the field. But because it's a small community, I want them to feel connected with me. So oftentimes I just bring them to my house. My wife is there. My 3 kids are there. They feel like part of the family, and we're throwing cast nets. We're fishing. We're going out in the water. We're exploring the marsh. All sorts of fun things. So I like that sort of personal aspect that's going on there. Because of that large tidal surge, we can go tide pooling. Not West Coast tide pooling, East Coast tide pooling. So what does that mean? Well, at high tide, you don't see any of these sandbars, but at low tide, these sandbars are exposed and they trap corals. You've got Lepidorgia, whipped corals, sponges, crabs, all sorts of cool things. Not as cool as the West Coast again, but nonetheless, really cool tide pools. The only bad thing is these sandbars shift constantly because of the movement of the water. So we're always getting stuck as we're just motoring through the marsh. We hit these sandbars and we got to wait for the high tide to come back in so we can get off of them. But nonetheless, we spent a lot of time exploring. Also, huge natural oyster reefs in the area. So when we're not out there studying them, we're literally eating them, which is a lot of fun. Absolutely delicious. Lots of fossilized shark's teeth in the area. So if we're running a lab and we're looking at tide pools but we're not finding anything, everybody get a shovel and let's start digging. So lots of megalodon teeth, and then the students go and sell them and make some money on the side, which is nice. Lots of barrier islands in the area. Some are state parks. This would be Hunting Island State Park with the characteristic lighthouse, the most popular state park in South Carolina. Why are we on the rock jetty? Because besides oyster reefs, there's really no hard substrate for things to settle on. So are we encouraging invasive species by putting these rock jetties out? Well, we're looking into that. And indeed, we're seeing range shifts, invasive species, all sorts of cool things. So we spend a lot of time on the rock jetties. And also some barrier islands are completely uninhabited, pristine. And so we spend a lot of time looking at this, the tree graveyard, looking at the trees, what's growing on the trees. And that's a lot of fun for the students. Although there are a ton of rattlesnakes. I just can't tell you how many rattlesnakes are on these barrier islands. Freaks me out. I just can't stand snakes. One of my favorite things to do is touch everything we come across and pass it to all students. All students have to touch everything. Look at it, oftentimes draw it, because I feel like you really don't understand something until you have to draw it. So we spend a lot of time picking everything up, assuming it's safe to do so. As I mentioned yesterday, my mission is to change the face of science, and so I specifically recruit Middle Eastern, Black, Hispanic, Latinx students into the lab, and had some successes, but most of the time I have failures, and when that failure happens is when they leave my lab and go on to graduate school. They're hitting a lot of barriers, a lot of headwinds, And the graduate students here gave me awesome feedback yesterday in terms of how to change that, things that I can do to help, help with that transition moving forward. I'm very thankful for all that feedback that I got. Having said this, there are white males in my lab, so I don't think that I'm biased. I am biased, but not that biased. But I do go after the diverse students and absolutely love it. So in New York City, I felt that you have a ton of diverse students here. Not finding that in the South, which is unusual because the general public is very diverse, but not necessarily in the academic institutions. But in New York City, it was easy to recruit. It was easy to inspire, especially at the museum with the location, with the views, with the behind the scenes specimens. It was really, really easy. And I got down to South Carolina and I initially struggled. I could get them into the lab, but I struggled to keep them interested. So I had to make a quick change, hence the shiny objects. Quick transitions. I already spoke about the fact that I spent more time commuting in New York City than with my family. So that was a big reason why I got up and left. So these are my kiddos. So I've got Jax, Ava, they're twins, about to turn 6, and Chloe, she's 2.5, absolute loves of my life. Spend so much time outdoors with them. Otherwise, I would be in the car commuting, but now I think it's maybe 6 minutes biking to work, which is absolutely fantastic. So I can spend all that other time with them. They absolutely love the crabs. They're all holding a green invasive, or the invasive green porcelain crab. It's one of their favorites, but every day we're out on the water exposing them to the environment, which is a lot of fun. So why the deep sea? Well, I want to tell you why I love the deep sea. Number one, the toys that we get to play with to go down, such as the Alvin. Yes, it's $65,000 a day to use this toy, but it is a ton of fun to go down in this and send students. That's been cool. The research cruises, which are fun, but they've also been a bit scary. So I'm going back a bit. So 2011, we were on the Polar, left out of the southern tip of South America, crossed the Drake. I hear it's either totally calm or ferocious. Well, we went out on a ferocious cruise, and the seas were just horrific. And so we were getting rogue waves coming over the side as we were out there trying to deploy some gear, because we wanted to be one of the few groups that actually sampled in the Drake. And so we're trying to get gear over. Well, then we hit a monster storm. So this is the back of the ship. Here is the A-frame. This is a wave about to approach. This is the wave starting to come on the back of the ship. And a lot of our gear, unfortunately, we were trawling. I hate trawling, but sometimes that's what you got to do. But a lot of our gear was chained down to the back deck, and there was a lot of ice in the water, and it broke the chains and we lost some gear. A lot of the windows were blown out, but thankfully we survived and got to tell the tale. But all of this being said, that it's not always rainbows and butterflies when you're out on research cruises. Sometimes it does get a bit hairy. Things get quite big in the deep sea. This is me with a little bit more hair down in New Zealand. One of my favorite things about being a coral biologist is you get calls from random people. They're like, oh, this is New Zealand, the New Zealand Institute of Water and Atmosphere. They called and said, a fisherman got this giant coral stuck in the net. You mind coming down here and tell us what it is? Well, it's already known, it's a bubblegum coral. I'm sorry. But things get rather large. I think this was anywhere between 12 to 15 feet tall, this particular coral that was bycatch in the fisheries, calcium carbonate chalk-based skeleton. Things get really old. This is a glass sponge from the South China Sea, aged at almost 18,000 years old. Like, what the heck? That's crazy. So quite big, quite old. I love, this is an actual figure from the publication. I thought that was just phenomenal. High heels in the publication. I thought that was super cool. Also more cool toys. And so I've been working with David Gruber at Baruch and Harvard Soft Robotics Lab to develop some underwater devices to hopefully indirectly sample deep sea organisms. So why? Because they're old. Not all of them, but a lot of them. And so we started with what we call the jellyfish tickler. This was a lot of fun. And so just gently rub these little fingers on the jellyfish, rub the DNA on the fingers, and then preserve those fingers, and you got their genetic blueprint. So that was fun. We published a paper on this in Current Biology, looking at collecting a jellyfish the normal way with like oil and gas manipulators that just squish the heck out of them and cut them in two, versus our jelly fingers, looking at the transcriptomic response. You know, ecology studies are really funny. You almost know the answer before you do them, but you still got to do them anyways. So obviously, if you just tickle the thing, it's not going to be as stressed as if you cut it in half, right? Well, that's what we showed, and we got a good paper out of it. And so in this case, we were just tickling it, only a few genes turned on, versus if we used a traditional manipulator, a bunch of OSHIT genes turned on, apoptosis genes and things like that. And anyways, there you go, ecology. And then fun things like this little sphere. You guys seen this? This is awesome. So, so things that are swimming in the deep sea, this little sphere collapses and you track whatever it is inside. You scan it and then you print this thing up on your ship. So you don't actually have to collect the specimen, but you get a, you can print out what it looks like. And there's also this little biomimicry beak that comes out and takes a little tissue sample and preserves it for DNA or RNA analysis. And then you just open this thing and let it go. Like, how cool is that? So, but having said that, I applied for the for a job, a curatorial position at the Smithsonian, which prides themselves on collections and having specimens. I presented this, and they're like, "Well, why would you use that? We need specimens." I'm like, "Oh, I probably should." Bad audience to be showing this to. And also finding such cool things like. So in 2019, we had a cruise to the Gulf of Mexico, Flower Garden Beach, National Marine Sanctuaries. We're only in about 100 to 200 meters depth on the shelf break. And we came across this thing for size. Here we got an arrow crab. Any idea what this thing is? So at this point, we were doing telepresence, so we were able to beam this on the internet. So people at home were weighing in what they thought it was. What do you guys think it is? Any thoughts? We call it the purple fruit snack monster because we didn't know. We didn't know. So we slurped it up with the ROV. It got on board and it opened up. It was a sea anemone. Had no idea before. And so we didn't even know this particular type of anemone occurred in the Atlantic. It's only known from the Pacific. And the one in the Pacific, these little structures are concentrations of the nematocysts. And the sister species is one of the most venomous sea anemones known. So thank goodness we didn't touch it. But we're currently doing the genome and transcriptome and looking at the venom genes, toxin genes in these particular structures. So that's pretty cool. And then this thing, Relicanthus. Just yesterday, I was on Twitter. Somebody thought they found another one. So this dates back, I think it was the 90s when the first specimen was found on the East Pacific Rise. Tentacles 10 feet long, mouth about 1.5 feet wide. And it was found around a hydrothermal vent. And at the time, they collected one specimen, got it back to the lab, Meg Daly at Ohio State, looked at it and said, "Oh, it's a sea anemone," and so called it Bolosauroides. Well, then we got another specimen in, and we did the genetics, and no, this is something totally new in the tree of life. Yes, a cnidarian, but we really don't know what kind of cnidarian. And so we renamed it Relicanthus daphneyi after, unfortunately, she recently passed, Daphne Fountain from Kansas State, sea anemone expert. Every time we get a new data set, this thing moves in the cnidarian tree of life. So full mitochondrial genome said maybe sea anemone. Almost 1,000 genes says that maybe it's sister to the black corals. We have no idea. So right now we're doing the genome, the transcriptome to see what the genome tells us about this particular thing. So just really cool creatures down there. Still a lot to be discovered. So how about black corals? My first love, my babies. They're called black corals because the skeleton is black. The tissue can literally be any color in the visible spectrum. We call them hotels in the deep sea because they are home to so many different things. This one colony had 2,500 other things living on it. So talk about important real estate there. That's awesome. So historically, these skeletons were collected and turned into amulets and scepters, ground into powder and consumed to ward off evil and injury, which is pretty cool. Hence, antipathies against disease is the first genus. This was a brooch that the Fish and Wildlife sent me to see if there were any black coral pieces in it. There weren't, but nonetheless, I thought it was pretty cool. They thought there were. Beautiful jewelry. I don't know if I mentioned this to some of you. My mom wears black coral jewelry. I'm like, Mom, if I can't convince you to stop wearing this, I might as well just quit. This is bad. I'm trying to conserve this because these are very long-lived animals. Yes, the jewelry is beautiful, but so what do we mean? So this particular species of black coral was aged over 4,000 years old, and it was only collected at 400 meters depth off of Hawaii. So perhaps things are even older than that. Some other black corals, some are 80 years old, some are 200 years old. So there's variability, but this seems to be the oldest one to date. I mentioned yesterday I'm working on a review paper on aging and longevity of deep sea critters. We need to talk about the whales. But yeah, if you guys are interested, if you think you have some new information you want to tell everybody, let's collaborate. Let's do this. I'm excited about this particular review. I think it'd be very timely. Also, not only black corals, the oldest corals in the ocean, they're also the deepest at 8,600 meters. Like, what is that? That's crazy. Most other corals stop around 2,000 to 3,000 meters. Black corals are like the honey badger. They don't care. They do whatever they want. And it's not just with regard to physical conditions, but molecularly, they're absolutely nuts. So here's what some black corals look like. This is one of the deepest black corals. It's called Abyssopathes because it lives in the abyss. Forms this little wind tunnel. Why? Because food is limited down there. And if you form this wind tunnel, kind of concentrate the food and grab it with your tentacles. We've got unbranched whips. We've got bottle brushes, both unbranched and branched. We've got bushes. We've got little sort of mini hedges, bushes. I don't even know. Green tissue on this one. And this is a good place to tell you why I think I can inspire students with black corals. Because every time we go down, we're finding new species. And guess who gets to name them? The students, which is really cool. So working in the deep sea, largely unknown, finding new species all the time has been very, very helpful. I mean, we've probably got 12 papers in the pipeline right now, all new species descriptions, all driven by undergraduates. So they're just sequencing typical mitochondrial genes, nucleoribosomal genes, putting them in phylogenies and then describing them, and also doing the morphology as well. So it's not just molecularly derived species. We've got feathers, stalked feathers. I mean, these are all different types of black corals, little mini palm tree-like things. Just so cool. These particular crabs aren't found anywhere else in the deep sea except for on this species of black coral. We don't see them walking around. We don't see them on any other black corals. Like, what the heck? Are these things, I mean, are there babies traveling together? We have no idea. So really cool stuff going on. So if you take that tissue away, you reveal the skeleton. It's a protein chitin-based skeleton. So we don't think it's going to be susceptible to climate change, ocean acidification, the same as your stony corals. But they're also called thorny corals because of all the spines on those skeletons. Spines come in all sorts of different shapes and sizes. Some are forked. Sometimes the idea of a spine, you're like, is this a spine? Is this multiple spines? Like, what is this? I have no idea. So my students and I like to debate what the heck we're looking at a lot. The polyps or the feeding mouths also come in different shapes and sizes. These are hexacorals because they have 6 tentacles. So you got the mouth and the 6 feeding tentacles and stinging tentacles around that mouth. Now here's where it gets a little hairy. So most things in the deep sea, they want to get up off the floor and get in that water flow to get the food and to get rid of their waste. And so things like to grow or live on black corals. You realize this isn't a black coral, this is a corallium. But nonetheless, they're doing this with the black coral. So what sort of things live on there? Well, parasitic zoanthids, we've got chirosterid crabs, brittle stars, tons of worms. My gosh, I'm literally covered in worms when we bring up these black corals. And I hope none are parasitic, and I hope I'm not filled with worm larvae or anything like. Anyways, lots of worms, which is really cool. But here's what happens with black corals. Their morphology, the way they look, changes based on who lives on them. So it begs the question. We've been, most black corals are in the deep sea, so 200 meters down to 8,600. Traditionally, we've used trawls to get them, but we've been describing them with no tissue, because by the time you get the trawl up, all the tissue's gone. We've just been describing species based on skeletons. Well, guess what? If what's living on them changes their morphology, have we been describing species based on had to say crapola characters? I think so. So that's pretty scary. So for example, if a particular worm gets on there, all these pinnules or side branches form a tube for that worm to live in. Well, we created a new family to account for this particular species that has this worm tube around, which I think is garbage. Anyway, so that's difficult if you're looking at morphology. Well, you might say, what about the DNA? Well, we got a problem there too. They mutate or change the DNA 100 times slower than everything else in the animal kingdom. Horrible. Now, this is largely based on mitochondrial DNA and nuclear ribosomal, but that's what we're finding. So we're screwed based with the morphology and the current markers. So that's been problematic. Just to show you how terrible it is, we collected hundreds of colonies of Antipathes grigi across the Hawaiian Islands, and given the currents through separating this series of islands from this one, we thought we'd see population differentiation. Well, guess what? We sequenced 5 mitochondrial regions, energetic spacers, CO1 barcode of life, and 3 ribosomal genes, ITS1, ITS2. Nobody even uses ITS. They do use ITS, they usually don't because it's so variable even within a single individual. Well, guess what? Across hundreds of individuals, not a single variable site. I was sickened years. I'm looking at the same DNA signature across all of these regions. It was demotivating, I tell you that. Then we've got this thing where, and this is the black coral tree of life known at the time, the genus Stichopathes was found in 2 different families, color-coded here. Well, that was weird. So what does that mean? That means historically, anytime we've seen this unbranched whip, we've said, oh, that's Stichopathes. You can't do that anymore. That's 2 different families that have converged on the same form. Yeah, what the heck is going on? Oh, and by the way, we just published on this, but I don't have it in here. The type species for the entire order of black corals was classified in the wrong family based on, so we're having to contact the zoological code and have all that changed. It's a big mess. Also, another mess. We've got 3 different genera for which I can't tell apart. Here's what they look like. We've got this big planer tree, this highly branched bottle brush, unbranched bottle brush, and this little crazy thing. They look radically different. I can't tell them apart using any genetic marker. And here's where they were collected. So this is the US West Coast, California, Alaska. And so we've got huge depth differences, the geographic distances, and I just can't tell them apart, which is absolutely ridiculous. So then I said, well, what if I find a black coral that's found worldwide and I collected it from different ocean basins? Would I find any differences? And so I chose Lafayette, this 4,000-year-old beast. There's only 9 species in this particular family. And so I collected 6 individuals from around the world, sequenced their mitochondrial genomes. Usually mitochondrial genomes are like 15 to 16,000. This one's a little bit bigger, so that's cool. More room for variation, right? So we got 1 from the Gulf of Mexico, 2 from Bear Seamount. So here you guys are on Long Island. 1 from Greg Seamount. So we've got, what is that? 4 from the Atlantic, 1 from the Mediterranean, it's a topo type. And then 1 from Hawaii, Pacific, right? So I'm like, we've got to be able to find some differences among these, right? Well, so here in the circles represent those 6 mitochondrial genomes, right? And so again, this is the differences in size. Well, across all 6 mitochondrial genomes, we only found 70 changes, which is not a lot, all right? Given this is a worldwide distribution here. And then of these 70, only 46 of these 70 changes were unique to any one individual. So let's show you where those changes are. So for this individual, there's only 1 unique mutation compared to these other 6. So of these 6, this one seemed to have the most number of mutations. 32 of the 46 were unique to this individual. Where do you think this one was from? You think it was Pacific, Mediterranean? What do you guys think? Mediterranean? That's what I thought. We did all this blind. Nope. So Gulf of Mexico, Mediterranean had 2 unique mutations. Hawaii had 5. And then within this, the seamounts, the underwater mountains just offshore, on the same freaking seamount, we had 1 with 4 mutations, 1 with 32. So I was like, oh man, what's the deal? So it's not geography. Is it depth? So I overlaid depth on these findings. It does, I mean, yes, these two are deeper, but they're kind of right next to one another. So I don't know. These all look like the same species to me. I have no idea what's going on with this one individual on the same seamount as this one. It doesn't have much variation. So black corals are just nutso. Then I met this guy, Jeremy Horowitz. He was a PhD student at the University of Townsville, the Coral Excellence Center in Australia. And he said, I'm interested in black corals. I said, well, stay away from mitochondria, stay away from ribosomal disease, go after ultra-conserved elements and exon loci and things like that. And so he did it. And he did such a phenomenal job. He just got a Peter Buck Fellowship at the Smithsonian in DC. Love this guy. He lives and breathes black corals. And so we've got a study that was just accepted into Proc B, where for 83 of the 200 something species, we had over 1,000 ultra-conserved elements or gene loci. And so we built a tree. There's variation there, so we can tell relationships. It has been awesome. So it's been a game changer for understanding the evolution of black corals. But the reason I want to share this with you is because this is neat. Well, I think it's neat. We thought that previously, based on a few groups, that shallow water taxa invaded the deep sea, and that's how deep sea organisms tend to be. However, what we're finding with the black coral, so we used a molecular clock and dated them back to over 400 million years, which is really cool. It seems like there's been big diversification events right after mass extinctions, which is sort of textbook examples. But what we found was that ancestral black corals seem to evolve on the slope. All right, they weren't on a shelf, they were not in the abyss. The ancestral black corals evolved on the slope, and only later invaded shallow water. It makes sense because shallow water, you've got sea level changes, you've got ice scour, you got all that other stuff. And then the abyss from time to time goes anoxic. So it makes sense that the slope would be like a refuge, if you will. So what do we see? Black corals evolve on the slope, then move into shelf areas, and only about 50 million years ago do they invade the abyss, which is crazy because most black corals live currently in the abyss. So it's actually a recent invasion into the abyss. So it's been a fun story to tell. And on top of that, we're trying to figure out morphological characters that would help them achieve these invasions, both shallower and deeper. It's been a fun study. So finally, we're getting a handle on the evolution of black corals. All right, leaving black corals behind, I've been forced to move shallow, and so we're jumping into oysters. Oh my gosh, I love oysters. This particular study is funded by the Spring Island Trust, in particular Chris Marsh and Andy Jones, the director. They're very interested in understanding our oyster reefs. What's an oyster? It's just a bunch of organs that taste delicious. That's what it is. And what's eDNA? eDNA is just any DNA that's floating in the environment. It could be because of sloppy eating, skin cells sloughed off, it could be feces, it could be any number of things. And that the DNA could be in a cell, it could just be hanging out, just naked by itself. And so all you literally have to do is take a scoop of water and sequence the heck out of that DNA to figure out who's home. And so we want to do that with oyster reefs. I hate text, but I just needed this to remind me of stuff. So I've got a million things I want to do with eDNA and oysters. Most of it is I want to figure out who's home. Who's actually living on the oyster reef? What do I mean by that? Yes, we know visually what might be living on there, but I want to know what parasites are living on there. What are small cryptic things that are living there that we can't see with a visual analysis? Down in the south, we close oyster reefs a lot because there's too much bacteria or outflow of sewers and stuff. So what happens to the diversity of oyster reefs when oyster reefs get shut down? Does diversity actually go up? Does it go down? I also want to know, as an oyster reef ages, how does diversity change? And so these are all sorts of things that we're trying to figure out using eDNA. So we've got lots of natural oyster reefs that we can go and look at. And so what we do is, we're not actually sampling the oyster reef for eDNA at this point, but what we do is we get in our kayak and we wait for the tide to come in. So the flood tide is coming in, and as soon as the tide covers the oyster reef, we take a sample of the water about halfway down between the height of the oyster reef and the pluff mud. I'll come back and show you how that happens here in a minute. We're also looking at artificial reefs. So we're working with the Department of Natural Resources, and so my students love building these things. So we build these cages, we fill it with a sterile oyster shell, or, which I'm not a big fan of this, plastic bags. We put more plastic in the ocean. Can you imagine? Deploying plastic bags, mesh bags with the oyster shell, all to aid recruitment of the spat or the oyster larvae. They like to land on their own. So we're doing that. So the students have been working with DNR in Charleston, setting these things out. And this is what it looks like. This is just a few months after deploying these artificial growth reef platforms. It's awesome how fast these things are growing. And so what we're doing is we're sampling this over time to see what sort of critters are occupying these particular reefs. Now, why am I taking this eDNA approach? Is that 5-minute mark? Oh, okay. Why? Because this is how we traditionally sample. It's like Godzilla coming on the reef, like, and setting down this quadrat, and we're trying to figure out who's home. Well, I bet you most things are doing this. Like, I'm getting the heck out of here. They're burying. You're like, what? Why would something stick around? And so I think our estimates of what's on there, I think, are off. And so I'm hoping eDNA will give us a better aspect, better look at that. So it's been since 1985 since somebody looked at the oyster reefs in the Beaufort area in South Carolina. And in there, it's all textual. And so what I had my students do is go through that text and find pictures of everything that's ever been found on the local oyster reefs. And the reason I'm having them do this is I want them to find a picture and be like, wow, this is cool. I want to learn more about this particular creature, hoping that we can get more taxonomists in our field. And it's been a lot of fun, all the different things that they've been finding. I have yet to have a student get interested in one particular thing, but I'm hopeful. I'm hopeful that that will happen eventually. So again, we're either waiting for the flood tide to come in and we take a sample. If we do, we use this horizontal sampler. All we do is we stick it down. Depends on the depth of the oyster reef, but we stick it down. So you've got plungers on either end. You send down this little messenger. It hits here, and the plunger closes. This is what it looks like when it's closed. And you get 2 liters of water. We only process one liter of that water. I had some reviewers who said, well, I don't like that approach. I think you should do something differently. You should also do the visual inspection. All right. So what we're doing is we're taking, this is in collaboration with Robert Dunn at Baruch. We're taking 5-gallon buckets, cutting off the top of it, filling it with sterile oyster shell, cutting out a hole in the middle of an oyster reef and keeping it there for 4 weeks. And after 4 weeks, we take it out, soak it for 30 minutes in seawater, and then we sequence the DNA of that seawater. And we also do a visual inspection of what's growing on this particular shell basket. And so that's been good. It's pacified a lot of reviewers moving forward. But you might ask, well, what's your negative control? Our negative control is we go out in the middle of the channel and we take a scoop of water there. And so if we find the same organism in both the positive control and the negative control, we compare the number of reads. So if I have 100 reads in my positive and 5 in my negative, I'm still going to keep it in my positive because that thing was potentially more abundant on the oyster reef. So once we get that water, we just filter it across 3 filters. This is marsh water. It is nasty. Not nasty. It's just goopy, and there's lots of stuff in there. So we do 3 different filters, 3 micron, 1.45, and we sequence the DNA across all of those. And then we send everything to GeneWiz for amplicon sequencing, targeting whatever it is that we're interested in: protists, bacteria, viruses, fungi, you name it, fish. And as I was telling the graduate students yesterday, get on social media. So I was promoting this project and the students doing it on social media, and somebody from bioscience reached out and said, hey, let's do a write-up on your project and your students. That was cool. I got an invited chapter to theoretically talk about what we're doing. So that's been cool. So, and all of this happened simply because I was on social media telling the world about what we're doing. So get on social media. Transition. How am I doing? 5 minutes? Oh my gosh, this is not good. Okay. And so leeches, why leeches? This is the Amazonian leech. This is about as big as they get. Love leeches. So there's 2 types. There's one that kind of Y-shape hole into you. There's another that just puts a proboscis inside of you. Why leeches? Well, it's been since the 70s that anybody looked at leeches in the Carolinas. And I love this particular quote. This species, Macrodella, is notorious for its tendency to attack man. Like, who puts that in a publication? I thought that was pretty cool. So they looked at it morphologically. Well, a student was interested in finding out more about what species live there today. So we're pulling them off sharks, skates, and rays, off of crabs, off of diamondback terrapins. We're pulling them off alligators. We're just having a great time. Most of the time we're doing freshwater, marine, and terrestrial leeches. Most of the time we're in ditches. You can see some of the traps that we set out, essentially putting liver and other stuff in there. Guess who's in the traps most of the time? Now again, I'm looking for leeches. Gators. Gators, and you guys know what amphiuma is? It's a legless salamander, 4 feet long, that loves to bite, and they're purple. Oh my God, I hate these things. They are aggressive. I can't even tell you. Every time I go to anytime I'm walking through these things, I really don't mind the gators. It's the amphiuma that I don't like. Nobody told me about amphiuma until I experienced one and said, what is that? I still can't stand these things. Snapping turtles, these things are amazing. I learned the hard way that even if you're holding them by the tail, their head could come out and get pretty close to you. So you got to watch out. But even get leeches living on these things as well. So we're plucking all these leeches off of the gators and everything else. It's been a lot of fun. Sea turtles. I think there's a correlation between the leeches on the sea turtles and HPV, which is interesting. So yeah, pulling off all this stuff. And what we're doing is we're just doing basic barcoding. Who's home? What are the species? We're also looking at who they've been feeding on. So we take out their blood meal and sequence all the DNA in that to see who they've been feeding on. And then we look at their anticoagulants to see, because they're secreting anticoagulants in you, but also into them, so that blood meal doesn't harden. So we're looking for novel anticoagulants. So that's been a fun project for the students. And then I've got to race through this. Horseshoe crabs. This is neat. So in South Carolina, they're the biggest out of all the US, the biggest in South Carolina. They generally come to, right now, in fact, I'm missing it. Right now, they're coming up onto the beaches to do their lekking, their mating events. But it usually happens as the sun is going down. So you really don't see a majority of the mating happening, because it's happening at night. Once they're done, beach is covered. All of this is all the eggs, fertilized eggs they left behind. Beautiful trilobite larvae, just the cutest little babies in the whole world. Love these guys. Unfortunately, a lot of them are getting eaten. But here's the problem. These red Nazi birds that go from North Pole to South Pole, they usually stop at 1 or 2 places. One is in the low country where I am to eat all those eggs. And then they head on southward. Well, the environmentalists are upset that this is happening. The fact that Charles River Lab is bleeding the horseshoe crabs for their blue copper-based blood. Why? Because there's amoebocytes in that blood that coagulate around bacteria. Why is that important? Anytime you've had a shot, the syringe, anytime you've had, you guys are young, an implant, anything like that, that has to be tested. Catheter that has to be tested with the blue blood of horseshoe crabs to look for contamination. So this is hugely important for our well-being and our health. It's very, very important. But at the same time, they have to collect these horseshoe crabs. They collect about 500,000 a year to bleed. But the environmentalists are worried, well, if you're taking all the horseshoe crabs, then the red knots don't have anything to eat. Well, the environmentalists were successful. They shut down all 30 islands where collections were happening in South Carolina as of like 3 weeks ago. So Charles River Lab is doing something a little bit differently. They're moving into the marsh. Why? Because we recently figured out that there's more horseshoe crabs in the marsh doing the reproduction than on beaches, which is pretty interesting. And here's a picture of them at night. So why am I telling you this? Well, Charles River Lab reached out and said, well, I need your help. You and your students, I need your help figuring out how many horseshoe crabs are offshore. How many are we working with? Well, Fisheries Council, the Atlantic Fisheries Council said it's a good number. What does that mean? A microsatellite study many, many years ago, almost 10 years ago, said the effective population is big, but nobody knows how many. Well, I submitted multiple proposals, nothing was funded, and I got frustrated, and I said, we've got to do this, we've got to figure it out. And so then I thought, let's use drones to figure out how many individuals are at least reproducing, and then try to get an estimate on number of individuals possibly in the population. It's got to be better than good. And so, horseshoe crabs are related, they're arachnids related to ticks, spiders, and scorpions and stuff. Well, you can use thermal imagery to see, remember, these are not endotherms, these are ectotherms. So theoretically, you shouldn't be able to pick them up. But I am confident that we're going to be able to. So the lower left are crabs, tick, scorpion. And so here's what I'm hoping. I haven't tested this. I got funding. I've got the drones, but I haven't even tested it yet. So here's the hinge. So we got the epistosoma, the prosoma, we've got the hinge. Well, right underneath the hinge is the heart. So you got a lot of hemolymph traveling through there. Also, in the heat of the moment, you've got to be making heat, right? I mean, if you're in a big pile having sex, I would hope you're making some heat. So I'm hoping that the combination of the heart, them having sex, I'm hoping we're going to be able to pick all this up. So private donor bit, we've got multiple Teledyne FLIR thermal imaging drones, as well as phantoms, an older model of phantoms, so that we can be looking during the day as the sun is going down, as soon as the sun goes down. So we can be counting during the day, and then we can see how many individuals are coming up at night. So that's where we are with the horseshoe crabs. Students are very, very excited. And I realize I'm totally out of time, aren't I?
You're okay.
I want to extend an opportunity. I mentioned this before, but we've got this uninhabited, pristine barrier island. Our campus is way up there. This is all salt marsh. It's called Pritchard's Island. Governor McMaster promised us $1.3 million to buy boats and things, 55-foot catamaran, blah, 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 sensors, all sorts of equipment, and then 600,000 reoccurring every year to study this island. I don't study barrier islands. I need you guys. I need help. So if you guys are interested, we're going to have a lot of money yearly to study this island. I've already spoken to some of you about potential projects. So please, I need help studying this island. What I'm going to do in the meantime, until I hear from you guys, so I'm going to send my students with their cell phones, and we're going to be characterizing all the plants and fungi and animals on the island. But I also need to look off the island. We're also going to be doing neofauna because this beach has never been restored versus like Hilton Head, where the beach has been restored. I want to see how that impacts the meiofauna community. So we're going to be looking at that, deploying haiku boxes so we can figure out which birds are stopping over. This thing has just been a game changer. My wife and I love the bird. This has been really cool. It uses neural networks to listen to the songs, records them, tells you who's home. That all feeds into the ornithology lab in Ithaca, New York. Some other things, we're doing sea anemones. So Piper Davis and her crew absolutely love sea anemones. So we're collecting all sorts of sea anemones. Your invasive one that you have here, Diutame lineata, the green orange striped anemone, they're all over the place. Bunadosoma, these upside down hanging ones, I don't even know what they are yet. Really, really cool sea anemones occurring in the low country. We're also looking at ctenophores because a student wants to look at ctenophores. Let's do it. So we're doing the diversity of ctenophores molecularly. Somebody was interested in transcriptomes of eels, so let's do it. Well, this is a bony fish. It's lacking scales, has a crazy microbiome in the mouth. How does it not succumb to the microbiome? Well, let's figure out if there's any cool antimicrobial peptides and stuff in there. We found a ton in the transcriptome. So just trying to keep students interested by letting them chase their dreams. I also want to throw out, we got 100,000 for this device, the MinION. MinION itself was like 5,000 to 10,000, but we had to buy 80-something flow cells at about 1,000 a piece right away. And I don't have enough projects to throw at this. So please let me know if you want a genome, if you want a transcriptome, let me know. We'll throw it through here. The longest read length is about 4 million bases, but on average, a couple 100,000. So please do let me know. Last but not least, I promise this is the last slide. And I want to plug David Gruber again at Baruch. Love this guy. He's just a brilliant individual. So he's doing a sabbatical at Harvard, and he was right next to the AI lab. And he went over to them and he said, you know what? Do you think we can speak with or understand the language of sperm whales? And they said, let's try it. So Project SETI came to be. This thing is so well funded, I can't even tell you. It's just, I've never seen money like they're getting. Essentially, they used AI to, you can see TED Talks on this, decode the language of sperm whales. And they're now deploying hydrophones and speakers and stuff like that down in off of Central and South America to speak back with them and try to have conversations, if you will. So if you don't know about this, check it out. It's going to potentially be a game changer. That's projectseti.org. This is neat. It's being driven by somebody right down the street from you guys at Baruch. I can't wait until things become public in terms of what they're finding. It is just awesome. With that, I will take any questions. Thank you guys for your time and the honor. I appreciate it. questions, concerns, anything? No? Is that boring? Yes.
I have a question. I was just curious about like how big a black coral individual can get.
How big? I would say Abyssopathes is probably this big, but the biggest one is maybe 10 to 12 feet that I've seen. Although I've read in the literature, some of those unbranched whips can get upwards of 20 feet long. I've never seen it with my own eyes, though. Good question. Yes.
So do you think they move towards the abyss or no one looked there and now you're just finding them, or you're discovered new sources, or?
So the ones that invaded the abyss have this unique branching to them, the pinnulation, the wind tunnels and stuff. So I think a species, ancestral species, was able to develop this new morphology so it could get more food in a food limiting environment. That's what we think. That's what we're proposing. Reviewers hit back hard, but we think we're going to get it through. Yes.
So black coral starting on the slope. Do you see any evidence of that in the fossil record, like in rocks?
So for bryozoa, we see that. For where was that? What's the other group? Bryozoans are the big ones that I'm remembering right at this moment. But it makes sense. I mean, again, the shallow water, you had huge extreme changes in sea level on the shelf, sometimes exposing the whole thing. And oftentimes the ice, the glaciers would come out and just rub it raw. And then in the deep sea, you have anoxic events. So where is that shelter, that refuge? Right there on the slope. Makes sense. So what the data are saying, you gotta follow what the data say. Yeah. Anything else? No? Yes.
When they're harvesting these horseshoe crabs,
Yeah.
concerned for the red knots, are they like harvesting them during an active breeding when they're on the edge of the beach? Because if not, then it's like, what's the difference of taking them out of the marsh versus the ocean?
So most of the year they're offshore, deep water. And then starting in mid-May, March, end of March, they all start coming to shore to do their mating thing. They were delayed this year because it's cold snap. They wait for the right tides, the right weather, and then they come on and they start doing the breeding thing for about a month and a half. And so as they're coming into the shallow waters, you get $2 per crab. So everybody's out there collecting these horseshoe crabs. Yikes. So yes, the females are much bigger than the males, and so they have more blood to give. But nonetheless, Charles River Lab still tries to collect most of the males, knowing that the female is going to be producing the eggs. And so they've got protocols in place to preserve the species, because without horseshoe crabs, they're done. It's $1 billion industry. And so, and I realize how important this bleeding process is. I feel horrible for the horseshoe crabs, don't get me wrong. Being strapped up with your telson all underneath and just getting bled. Oh, it sounds horrible. But from a human aspect, we'd be in bad shape without their blood. So I feel for the environmentalists, I feel for Charles River, I'm torn. And then the Defenders of the Wildlife called me the other day when they shut down these 30 islands. They called me and they said, we need your students to monitor whether or not Charles River is living up to what's going on, with regard to the court order. I'm like, my students, you want them to be between environmental firms and billion-dollar industry? So I thought about it a lot. I talked to a lot of people, and in the end, I didn't feel it was in the best interest of my students for their safety. I think there might have been some intimidation going on, and you had to work only at nights because that's when they're coming in. So I said no, and I recommended an environmental consulting firm to take it over. So I felt as much as I think it'd be great for my students, I don't want to see them get hurt or intimidated or anything like that. Two big powers. Good question. Yes.
Is there anybody looking into making something synthetic?
There is a synthetic, but it's only 76% effective. Which means what? 24% of the time you put a hip implant. Oops. So it's out there, but it's not good enough yet. Horseshoe crab gets it every time. There's word that Charles River Lab has a synthetic. As soon as that horseshoe crab industry completely gets shut down, they're going to release it. But that's just hearsay.
Were you using the mid-ion to look at genomic differences in black corals?
Right now, I'm just feeding anything I can through it. So one of the students, being in the South, we have these big live oaks, called Spanish moss. They said, I wonder if the Spanish moss from this tree is different than that one. So we're doing genome sequencing of Spanish moss. So it's just whatever interests the student, that's what we're doing. So black corals, I have yet to put a single black coral through there, nobody's that interested. I'm interested, but put the student first. Yes,
You said that black corals are not vulnerable to ocean acidification.
as vulnerable as stony corals, just because they have that protein-based skeleton. Yeah, calcium carbonate will be affected well before the protein. At least I hope. I mean, I don't hope. I mean, I hope nothing gets affected, but yeah, you know what I mean, right? Anything else? No? Thank you guys again. I appreciate you being here. Thank you.
Okay, so there's lunch, there's pizza and Challenger 173, so we can all head over there and keep asking Dr. Brugler any more questions that you have. Thanks,
Speaker
guys.
reads first, please.
Yeah, speaker reads first.